we're going to apply what we've learned in the last couple lectures to our biological molecules called carbohydrates. So these have the functional groups we've been talking about in the form of carbonyls and aldehyde groups or ketone groups or the OH groups, the alcohols, hydroxy groups. So these molecules contain those functional groups. So this is a great time to introduce them because you just learned about the molecules, so it's time to learn where they appear in biological molecules. So carbohydrates are the first of these you're going to study. Okay, so what is a carbohydrate? The name is a little misleading because when it was named, it was almost understood what it was, but not quite how it was put together. Right? So the name means a hydrate of carbon, carbohydrates, which means carbon with water molecules attached to it. It's a hydrate of carbon. And there were hydrates of other elements like metals and such, and it made sense to call it that at the time. Because if they looked at the formulas, they couldn't understand how the molecules were put together yet, but they could get a general idea of what the formula was, and you can do that chemically. They knew there was an oxygen for every carbon, and for every carbon there were twice as many hydrogens. So from that point of view, it looked like there was 2 hydrogens and 1 oxygen for every carbon. Well, that means there's a water molecule for every carbon, right? So that's what the nomenclature took off and called it a carbohydrate. But it's not actually true. A better way of saying it is for every carbon, there exists an H and an OH, which is almost correct. But if you look at that formula, something should stand out to you. Look at the number of carbons and the number of hydrogens. What stands out to you?
It's the same formula as if there was one site of desaturation.
Right, there's one site of unsaturation, correct. So because I don't have that +2 on the end, I must have subtracted 2, and therefore I have a site of unsaturation. That's exactly right. What constitutes one site of unsaturation? What are my options?
Either a ring or a double bond.
Right, but not both at the same time. So either a ring or a double bond. You're absolutely right. So for our carbohydrates, that's what we're going to find. And our double bond would be in the form of that carbonyl we talked about. Right, so it's either going to have an aldehyde or a ketone group there, or it won't have those, and it'll be in a ring form. Right, so we'll see how that happens towards the end of the lecture. Right, but every carbon will have an oxygen attached to it. And that's what it means to have CNON up there. For every carbon, there is an oxygen. Right, so we're going to see that most of those are going to be OH or hydroxy groups, right? And then one of them will be double bonded in the case of an aldehyde or a ketone. Okay, so to remind you that, let's start at the bottom with a simple 6-carbon molecule called hexane. Right, it should not be trouble to name and visualize at this point. This is trivial. Right, and then on the hexane, I'm going to place a carbonyl at the end. This does not make the chain longer. Right, I did not add another carbon. I simply added an oxygen. I didn't turn carbon number 1 into an oxygen. That would have shortened the chain. So I just added an oxygen to it. This makes it an aldehyde. How do I know it's an aldehyde? Well, there's a double bonded oxygen there, and it's on the end. So therefore, there is a hydrogen still bound here. Right? And then on every other carbon of this chain, not number 1, but every other carbon of the chain, numbers 2 through 5, I'm going to put hydroxy groups on it. Right, so I put an OH on every carbon except the one I've already put an oxygen on. So this has 6 carbons, as we started with, hexane. It still has 6 carbons when I'm done, and now it has 6 oxygens. Right, and if you want to count the hydrogens, you can, but it's unnecessary. You can figure it out from the formula. There is one site of unsaturation there, and that is the aldehyde at the top, so therefore we have 2N number of hydrogens, or 12. Right, and that's the case for every one of these molecules. All 3 of these have the same number of carbons, and the latter 2 have the same number of hydrogens, because this one doesn't have that double bond. Right? This one would have 14 hydrogens. Right? So this is what your sugars will look like. It's going to have an aldehyde or a ketone somewhere in the molecule. Of course, the aldehyde will be on the end by default, and the ketone will always be, for the sugars, on number 2. Right? It can't be on the end because that would be an aldehyde, but the ketones will always be on number 2 if it's a ketone sugar. right? They won't be anywhere else. No matter how many carbons it has, it'll always be on number 2. Right? So aldehydes are always on number 1. Ketones for sugars, for carbohydrates, will be on number 2. Every other carbon will have an OH on it. Does that make sense so far? Every carbon will have an oxygen attached in one of 2 ways. Either an OH, as in the case of most of them, and one of them will be a carbonyl. Aldehydes, it'll be on 1. Ketones, it'll be on 2. Okay. We have a name for this. We could have called it, 2, 3, 4, 5, 6 pentahydroxy, because there's 5 hydroxy groups on a hexanal, but it's also known as glucose. Right? So yes, you should be able to name the IUPAC name. It's not that difficult building it up like this, right? It's a hexanal with 5 OH groups. And yes, they could have been elsewhere, so I need the numbers. But we simply call this one glucose. Okay, so I want you to learn these sugar names like glucose and maltose and sucrose and all the others we're going to talk about today. And I'll give you a list at the end. Okay. So let's talk about instead of the aldehydes, the ketones with multiple hydroxy groups. So again, we'll start with hexane. Okay, this time I'll make it a ketone, but of course I won't put it on the end. I'll put the oxygen on the interior carbons. And I said for the carbohydrates, it'll always be on number 2. So this is 2-hexanone. And then if every other carbon, numbers 1, 3, 4, 5, and 6, I'm going to put a hydroxy group. So there they are. So they're on 1, 3, 4, 5, and 6, pentahydroxy 2-hexanone. Right? This also has a common name. It's not glucose anymore. It's the other major sugar we're going to talk about. And this is fructose, right? Or fruit sugar. Right? So those are 2 of the sugars I definitely want you to know. They're both 6 carbons. They're isomers of one another. Right? If you remember what that word means. They have all the same parts. They're just put together differently. Right? One's an aldehyde, one's a ketone. Okay? So let's look at some other definitions for carbohydrates. If there's only one sugar present, and we'll define what a sugar is in a moment, more specifically, if there's only one sugar present, like the ones we've seen, glucose and fructose, we sometimes call it a monosaccharide. Right? And saccharide and carbohydrate and sugar are 3 words in 3 different languages that mean the exact same thing. We tend to use them for different purposes, like carbohydrate is used when you want to talk about nutrition generally. The word sugar is just a more generic term for these things, and a lot of people don't even know what sugar means. They misinterpret it as other things. And we tend to use the language, the saccharide term, to indicate the polarity of it, or the how many of them there are. So if it's a monosaccharide, there's 1. If it's a disaccharide, there's 2, and so forth. Oligos means a few. It's an undefined number. Or in polysaccharide means many of them, right? 10,000 or so of these sugars put together. And you probably can think of a few of these. We talked about glucose and fructose being monosaccharides, and there are plenty more, like galactose and mannose and other things. Disaccharides. There are a couple I want you to know. We're going to talk about a few of them. For example, sucrose and lactose are 2 examples. We'll talk about a few more, like cellobiose and such. And then there's some that are a little longer, like trisaccharides. We're only going to briefly mention that they exist, like maltotriose, but we won't talk about them too much. And then polysaccharides you're familiar with, you just don't realize it. Things like cellulose or glycogen or chitin or amylose. All these are polysaccharides, and there are many, many, many sugars together. And a lot of misinterpretation of the word sugar, they interpret it as a synonym for sweet, and that's not true at all. Not all sugars are perceived as sweet, and many of them, such as the cellulose and chitin, aren't detected as sweet at all, but they're made of carbohydrates. Right? So these are one of our building blocks of all the biological molecules we have. There's 4 different types, and we'll get to them as we go along, but the first one we're going to talk about are the carbohydrates. And so we have single carbohydrates, some that are put together in short chains, like oligosaccharides, maybe 2, 3, 4, 10, 20. Right? And then polysaccharides, which are many, many carbohydrates put together that make up more functional units. So like the cellulose and the chitin are structural units for those organisms, for plants and crustaceans. Right? Your other least favorite word we're going to go over now, we talked about isomers before, and I know a lot of people don't like those words, isomer, but the other least favorite word you're going to have is chirality, if a molecule is chiral or not. Right? So when you look at an object, any object you want to compare, and you compare it to its mirror image or its mirror reflection, you have one of two choices. Either the mirror image is exactly superimposable on the original, like a basketball is exactly superimposable on the basketball's image, you wouldn't be able to tell them apart, or they're non-superimposable, like if you hold up your left hand, right? If you hold up your left hand, look in the mirror, it doesn't really look like your left hand anymore. It's reversed, right? So it has a chirality to it. It's left-handed, whereas the mirror image we'd call right-handed, right? I know that's a silly example because it's actually a hand, but molecules behave the same way. If a molecule has a mirror image that is identical to its original version, then we call it superimposable, right? And that molecule can't be chiral. If a molecule has a non-superimposable mirror image, then it has to be chiral. It has something in it that makes it left or right-handed. Okay, so let's look at some examples below, and I'll start with the easiest and simplest ones, and probably the only ones we're going to do for this course. Is if you have a carbon, right, or an atom with 4 bonds, and we're generally going to talk about carbons, and if it has 4 single bonds, the geometry was tetrahedral, right? Everyone remembers that, right? 4 things attached. It gave you 4 Vesper groups, no lone pairs, tetrahedral molecular geometry. In the case for that, if it's going to be chiral, all 4 things must be different, right? No 2 must match. So if you look at some examples below, we have methane here, right? In the case of methane, do any 2 of the 4 things attached to the carbon match? Yes, in fact, all 4 of them are the same. Didn't have to be, but if any 2 match, then it is not chiral. So that narrows our view of which ones can be chiral significantly, right? Chloromethane, again, not chiral because at least 2 of them match. Chlorofluoromethane, right? Again, at least 2 of them match. It doesn't matter where they are, but 2 of them are the same, so it is not chiral. I can rotate this to make it match any mirror image of itself. However, the 2 at the bottom have 4 different things attached at that position, right? So this one has a chlorine, a fluorine, a bromine, and a hydrogen, as does this one. But in no way can I rotate the one on the left, rotate it any way you'd like, right? Flip it around in 3 dimensions, whatever you'd like to do. There is no way to rotate it and make it fit exactly on top of this one. There'll always be a mistake, right? Because these have a chiral center, right? They have that center that has 4 unique things attached to it. And so it's either right-handed or left-handed, right? The one on the right here we'll call right-handed, so it gets a little R. And the one on the left we'll call left-handed, which gets a little S. S stood for sinister, which meant left-handed originally. There are other systems besides R and S. There's E and Z and D and L and so forth. But they all mean the same thing. I'm not going to get stuck on any one system for you. But they all mean the same thing, that there's 2 versions of it. There's a mirror image of it and the original image, right? Our right and left-handed chirality. That's what this means. Do you have to know which one is which? No, not for this course. But I want you to recognize when it's possible. Okay, so we will talk about some D versus L in a few minutes when we get to the sugars, but when I just show you a molecule like this, and we'll do an example in a minute too, all I want you to say is that this center, this particular carbon, is a chiral center. So therefore there's 2 possibilities. There's the left-handed and right-handed versions of it. Which one's which doesn't matter to you. The fact that you can recognize that there are 2 is all I'm interested in. Right, so let's do an example. So here's a molecule I drew below, and I want you to find all the chiral centers in this molecule. So I'm going to give you some algorithmic steps to go along the way. So the first thing we need to do is identify all the tetrahedral centers, because if you're not tetrahedral, you can't have 4 single bonds. So some of these we can eliminate right away. So the carbon right in the center, there is no chance that that's chiral, and you can eliminate it instantly because we know there's not 4 single bonds, right? There's a double bond there, so there's no way this carbon is tetrahedral, right? So we can eliminate that right away. So of the other carbons there, we need to identify all the atoms with tetrahedral geometry. So all the others clearly have 4 single bonds, right? I'm going to circle them here for you. But they don't necessarily have to all be one thing or all different. We don't know that yet. I just know those have 4 single bonds because there's clearly no double bonds present, right? Or triple bonds or benzene rings or anything like that. So each of these has 4 single bonds. Some of them have more than one of the same thing attached. And the easiest thing to see would be hydrogens, although you physically can't see them. You know which ones are CH3s right away. You've drawn enough of these to figure out the ones on the ends are definitely CH3s. So we need to eliminate all those that don't have 4 different things. So the ones at the ends of these lines, they're definitely just CH3s, right? They can easily be eliminated. What about the one on the right here? I see 2 methyl groups attached to that point, right? 2 identical things attached at the same carbon, so that one is definitely not chiral. The one on the left, however, has 4 unique things attached. And let's try to figure that out. All right, so the one on the right here definitely had 2 of the same thing attached, so we can eliminate it. The one on the left has 4 unique things. It has this OH down below, and I don't just look at the next atom, look at the entire group attached. So there's an OH down here, there's a methyl over here, there's all of this attached at this point, and there's a hydrogen not drawn. None of those things are the same, right? They're all different from each other. So therefore, this carbon is chiral, right? So I'll go through that one more time with you to make sure you get it, and then we'll do some examples. So we're going to identify all the carbons that have 4 single bonds, right? So I see all 5 of those, and then I want to eliminate those that are clearly have multiple things attached that are the same. So the 3 CH3s can go away, right? Because they have at least 2 things that match. The carbon on the right has 2 identical groups attached, those methyls, so that's not chiral, we can eliminate it. And the one on the left has 4 unique things. There's the methyl group to the top left, there's the OH down below, there's this entire rest of the molecule attached over here. I'm not just looking at this carbon, I'm looking at the whole thing. It's different from any other thing. And there's a hydrogen not drawn, which is different from all the others, so this one is definitely chiral. So this molecule had 1 chiral center. Okay, so there's a left-handed version of this molecule, and there's a right-handed version of this molecule, right? I don't know which one is which from the drawing, because you can't tell which way it goes, but it's definitely possible. That's what I want you to get out of this. There is 1 chiral center, and you can identify the carbon to which that center belongs. Okay, so here's another one I want you to do with me. So the first step is identify all the carbons that are tetrahedral. Okay, so identify all the atoms with tetrahedral geometry. Well, these would be all 6 carbons because they're all 4 single bond attached to them. Right? It's not a benzene ring. This is not a flat plane. This thing will adopt a chair in boat conformation and so forth. But you need to see that those are all 4 single bonds. Some of these carbons can be easily eliminated with less than 4 unique things, and that's going to be these 3 CH2s, right? So these all have 2 hydrogens easily, right? So we can eliminate them. They have 2 things that match. The remaining 2 carbons have 4 unique things attached, and let's make sure we see how that's the case. There obviously is a hydrogen here, right? Because it needs 4 bonds. So hydrogen is unique from everything else on this carbon. Fluorine is unique from everything else. And as long as these 2 aren't the same, then it's a chiral center. Well, so far they're the same, but if I keep going, they don't match anymore. And you have to keep going until you find a difference or you run out of things to check. Remember, I'm checking everything attached over here. So if these were symmetrical all the way around, as if, in other words, if this fluorine wasn't here, then this would not be a chiral center. But because of that fluorine, it makes a difference, and this makes this chiral. Similarly for this one, if I go around, they start to match at the first position, but then they differ here. Right? So because all 4 things are different, this one's also chiral. All right? Try this slight variation on the right. Let's follow the same rules. Let's identify all the atoms with tetrahedral geometry. Well, that's going to be all 7, because I don't see any double or triple bonds in here. Right? So all 7 are tetrahedral. The methyl group sticking off to the right and those 3 CH2s definitely are not chiral, so we can eliminate those. How about that one in the middle? Is it chiral or not? Are there 4 unique things? No. This is a methyl group. There's a hydrogen here. And these 2 things going around from this carbon are identical to each other. Right? There's no difference between these 2 going around. There were differences over here. There's no difference here, so that makes this not chiral. However, these are still chiral because there's a difference as I go around. This has a methyl on it, this one doesn't. Right, so that's how you find these differences. Keep checking until you find a difference or you run out of atoms to check. Right, so one more, a realistic real-world example. Here's a molecule. It's an artificial sweetener called NutraSweet. The actual name for it is aspartame. Right? It's made from 3 amino acids stuck together, which we'll talk about later when we get to amino groups. But you can ignore the nitrogens for now because they're not chiral centers. Let's look at the carbons. How many chiral centers does NutraSweet have? Well, let's take the molecule and follow our rules. So identify all the atoms with tetrahedral geometry. And you should find there are 5 of them. Right? All these carbons with 4 single bonds. Why am I not looking at these carbons? Because they're involved in a double bond. Not because it's to oxygen, but because it's involved in a double bond. It can't have 4 single bonds. Equivalently, all the ones in this benzene ring down here can't have 4 single bonds because they're involved in double bonds. So ignoring all those, and nitrogen only has 3 bonds, right? So it can't have 4. So ignoring all those other options, and of course oxygen only has 2, there are 5 carbons left to consider. Right? So of these, can you eliminate any of them quickly? Right? Before I hit the button to make it happen, can you eliminate any quickly, and what are your reasons?
The one on the left end has 3 hydrogens, so that one could be eliminated.
I agree. Any others? These will be the easiest
The
ones.
one on the right.
Right, this is the easiest one to pick out, I agree. So one on the right, this one, why?
Yes. It has 2 hydrogens.
It has 2 hydrogens, good. Any others?
The one attached to the benzene ring also has 2 hydrogens.
Right, so does that one. So we can eliminate the methyl group, right, rather easily every time. You'll see those right away, so you won't even consider them anymore. And these 2 were both CH2s, yes. How about the remaining 2? So this one has a hydrogen on it. It's got this big benzyl group on it. It's got this group with the oxygens on the left, and it's got the rest of the molecule to the right. All 4 of those are different things, so this is a chiral center. Equivalently, this one has a hydrogen on it. It doesn't have to have a hydrogen to be chiral. These examples all happen to. Right? A hydrogen, it's got an amino group up here, which we'll learn about later. It's got this oxygen-containing group on the right, and it's got this massive group on the left. Right, so all 4 of those are different, so therefore it's chiral. So how many chiral centers does aspartame have?
2.
2. Does that mean this molecule has a left and a right-handed version? It's kind of hard to describe, doesn't it? It's got 2. What do you do with that? When it was 1, it was easy. It's a left or a right-handed version. Well, each chiral center can be left or right-handed. So how many versions of NutraSweet are there?
Is it 4?
4?
It's 4. And how did you get 4? Tell me the math behind that.
Because each center it can either be left or right.
Right? And what's the math
So-
behind getting to 4? I know it's a silly
I don't
question,
know.
but there are 2 ways to get to 4.
Squared?
Say again?
Would it be 2 squared?
It's 2 times 2, exactly. It's not 2 plus 2. That's incorrect. It's 2 times 2, or 2 squared. So it's 2 raised to the power of the number of chiral centers you have. What if I introduced a 3rd chiral center? How many versions would there be?
9.
So 2 times 2
8.
times 2 is 8. Right. Because you don't change the base number, it's still a 2. You change the power. So it's 2 to the 3rd power. So every time I introduce a new chiral center, for instance, if I added a 4th chiral center, there'd be 16 combinations here. Right? So it's like flipping coins. How many ways can you get all heads or all tails, right? Or how many different ways can coins come up? If I add more coins to the list, I double my options. And that's what's happening with these chiral centers. So if you were a chemist working to make an artificial sweetener like this, and you plan to mass produce it and sell it, the fewer chiral centers you have, the more efficient, the less expensive, and the less time it takes to actually synthesize the right molecule. Because if you put the wrong version of these left and right-handed chiral centers, it's not sweet. Only this one is sweet. where it's a particular combination of one's left and one's right. Right? They can't both be left or both be right or the other way around. So only 1 out of 4 molecules made is actually sweet, and you have to purify that because the others are not sweet. They're not useful for this purpose. So if I introduce a drug, not necessarily NutraSweet, but another drug that has 9 chiral centers in it, it's going to be nearly impossible to purify without losing 90+% of my product. It'd be very wasteful to try to make that. So if we eliminate chiral centers as much as possible, sometimes you need them, but if we eliminate them as much as possible, it makes the drug easier to produce, it makes it cheaper to make, which makes the cost of the drug less. Right? So you see, this is a problem when you're synthesizing things chemically, is you don't always get the right product. You can get one of 2 possibilities for every chiral center. And a way around that is to use enzymes to make it, because as we'll learn later, the enzymes always make the same product every time, not a half and half like Markovnikov's rule type stuff. Okay. All right, so on to your other favorite word, isomers. It's coming back. So I talked about the ones in the top before, right? The ones in the blue box here, the constitutional isomers. You remember that definition. It was they have the same parts. That's true for all isomers. And the parts are connected differently. If they're connected differently by way of their carbons, right? If the carbon skeleton is connected differently, we call them skeletal isomers, which are still constitutional. It's just a subcategory there. If they're connected identically among their carbons, but something else is connected differently, like hydrogen or oxygen or nitrogen or any other element, right? We call them positional isomers, anything but carbon, right? And both of those are still constitutional. And that is the group of constitutional isomers. We also talked about functional group isomers, which are kind of off to the side there in green. It's kind of a what color is your car type thing. They're not necessarily a group among constitutional. They're a separate question. Like, do you have different functional groups? That's all that matters. And then the one below is the new one that you haven't seen before. You have seen it, but we haven't discussed it. Right? So these are called stereoisomers. And the idea here is they have all the same parts again, but they're not connected differently. So they're not constitutional in this respect. They're connected in the same way, but they go about it differently. So there's a left and a right-handedness to it, perhaps. All the same parts are there. They're all connected to the same carbon, but it's left or right-handed. So that's our stereocenters we just introduced, right? Our chirality. So if it has one chiral center, like the ones we've been describing, not NutraSweet, but some of the others, then we call them enantiomers. Right? So this enantiomer means it's one chiral center. It's a non-superimposable mirror image. Right? That's all that means. Okay? If you have more than one chiral center, then we call it a diastereomer, which makes sense. It means 2, right? 2 stereocenters instead of 1 stereocenter. And 2 stereocenters is one way to do that, or 2 or more, have multiple chiral centers. But diastereomers also encompass a group that you've already seen, and these are the cis-trans isomers. When you had your double bonds or you had your rings that were cis or trans possible, these are grouped into the diastereomer category because they don't have a stereocenter, but they do have a left or right-handedness. There's 2 versions of it. There's a cis and a trans. Okay, so those are grouped into the diastereomer category here. Right, so remember this little chart, right? You got constitutional up here, which are something's connected differently, if it's the carbons or if it's something else. And then you had stereoisomers, which still has same parts. It's still an isomer, but it has either one chiral center or multiple chiral centers, or they're cis or trans. Right, so these groups are all connected the same. They just differ in their handedness, left or right-handed, cis or trans, so forth. There's always a dichotomy of the 2 things. And if you have multiple chiral centers, like we just discussed with NutraSweet and others with more than 2 chiral centers, the number of stereoisomers grows rapidly, right? It grows exponentially. Okay? So let's go on to our monosaccharides, our sugars themselves, and start to classify these guys, that you've got this stereoisomer idea down. Okay? So the easiest way to classify sugars is to simply count their carbons. And you've been doing this since day one, right? Well, first thing we do with every molecule is count its carbons. So we can call them by their number of carbons. So if it has 6 carbons, we'll call it a hexose. This ose ending indicates sugar. Right? If you see ose as an ending, it means sugar. So a hexose or a pentose or a triose or heptose or octose or tetrose or whatever number it goes, just prefix the ose ending, and that means I have a 6, 5, 4, 7, 3, 8, whatever, carbon sugar. Okay, so if I showed you a picture of a 4-carbon sugar, it would be called a tetrose. Right? 6-carbon hexose. Makes sense, right? The other way to classify sugars is by their functional group. Right? They all have tons of OH groups, so that doesn't help us. But they're either going to be an aldehyde or a ketone, right? Depending where the carbonyl is. And they all have that carbonyl on either 1 or 2 in their straight chain forms. So here we're going to have an aldose, that means an aldehyde sugar, or if it's on number 2, it's a ketose. Again, that ose ending telling you what it is. And I can put these prefixes together. So if I have a 6-carbon monosaccharide, that means 1 sugar, so a 6-carbon sugar with an aldehyde group, it is a hexose, and it is an aldose, right? 6 carbons, aldehyde. So I put them together and called it an aldohexose. That tells me everything about it. It's got 6 carbons, and it's an aldehyde sugar. So aldohexose. Another example would be a ketone with 5 carbons, would be a ketopentose. So this should be a very simple system. You have 2 choices for the prefix, aldose or ketose, and then just list the number of carbons and append it with ose. So aldohexose, aldoheptose, ketotriose, whatever you come up with. Right? There's a combination to describe the sugar. And then in each one of those sugars, we have to decide how many there are in that family. And that comes down to our stereoisomers, or our chiral centers. Okay, so let's start with the simplest ones. Right, so I want you to learn a few biochemically important sugars. Right? We're not going to go through every one of them because there'd be hundreds. But we're going to do some ones that are important in biochemistry. Right? That's important in the function of the cells. So here's our important sugars that have only 3 carbons. And there's only 2 of them. There's the aldo version and the keto version. There's the 3-carbon version where it's an aldehyde, and the 3-carbon version where it's a ketone. Right? These are the only 2 that we're going to talk about, and that's it. There's really no other one to worry about. Every one of these is either going to be an aldehyde or ketone, as you see, so we'll have pairings like that. This one only has 3 carbons. Since it's on number 1, it makes it an aldehyde. It's on number 2, it makes it a ketone. And every other carbon that's not the carbonyl carbon will have an OH on it. On the one on the right, the ketotriose here, how many chiral centers does it have?
None.
None. So you didn't bother checking this carbon, right? It was a quick write-off. You went, nope, it's in a double bond, can't be chiral. These 2 required a little closer scrutiny, but then you instantly realized what?
Each of them has 2 hydrogens, so they can't be chiral.
Exactly. Right? So each of them has a pair of matching things, in this case hydrogen. They can't be chiral. So no chiral centers in this one. How about the one on the left?
The one in the center.
It has 1.
Right? It has 1, and it's this one right here in the center. Right? Carbon number 2
Yes.
is chiral. Right? The bottom one is not, again, because it's got 2 hydrogens. The top is involved in a double bond. We can write that one off. So the one in the center here actually has 4 unique things. It has an OH. It has this bottom group, which is different. We have this top group with the carbonyl on it. That's different. And it has a hydrogen. So all 4 are different. This is a chiral center. And we're only going to focus on the sugars that are D sugars, right? So that means if it has a chiral center, this one does, and it's the farthest chiral center from the aldehyde, which in this case is the only one, and we're going to say it's the D version, or the right-handed version of this one, is all that actually occurs naturally in the body. The L sugars do occur. I'm not saying they don't occur at all, but the D sugars predominate in biochemistry. Right? We're almost always going to use D sugars. Okay? So these are very simple to learn. 3 carbons long, either an aldehyde or a ketone, and every other carbon besides that one has an OH on it. Okay? That's very simple. Right? And the names for these, we called them, it was an aldotriose and a ketotriose, but the actual names are below, and you probably can write those if you wanted the IUPAC names, which I want you to be able to do. This is old news. But I want you to learn their biochemical names. The one on the left is called glyceraldehyde. Clearly, remember, it's an aldehyde. The glycer prefix means 3 carbons. So glyceraldehyde is a 3-carbon aldehyde with OHs on every other carbon. And over here is dihydroxyacetone. So you remember, if you cover up these 2 OHs, this is acetone right here. and it's got 2 hydroxy groups. Yes, we should label them 1 and 3 in the IUPAC name, which we have, but commonly in biochemistry this is called dihydroxyacetone, and I want you to remember that. Okay, so glyceraldehyde and dihydroxyacetone.
Are they also called aldotriose though? Do we have to know both?
When aldotriose is a category, right? It's a 3-carbon sugar that is an aldehyde. In fact, this is the only member of that category. They're going to get larger later. I picked the simplest one first. But this is a category, and it's the only member in that club. Same thing here. But when we get to more carbons, you'll see there'll be plenty more options. But yes, I want you to know everything on this slide here.
Question, would the D group always have 1 chiral center?
The D group, as you see, you're going to see, I know it's not clear at the moment on this slide, but you're going to see in the coming slides when we introduce longer carbon chains, it describes the last chiral center. Right, so hang on just a minute and it'll make sense.
I have a question.
Uh-huh.
Wait, if it's not a D group, like what is it? Like D opposed to what?
As opposed to L. So D is essentially right-handed, stands for dextro, which means right. And L is for left-handed, or levo, which means left. Right. So it's like
Wait, so what is R right-handed, or is it just D?
Right. So there's multiple labeling systems, like I said earlier. We have R versus S, we have D versus L, we have E versus Z, and there's a few others as well. But they all have that dichotomy, one or the other.
Okay, thanks.
So something that is D could also be R, could also be E, right? And something that's L could be S and could be Z, right? It just depends on your definition of that system. And I'm not making you learn any particular definition. We're just going to use D and L because that's what biochemists generally use when we talk about biological molecules. Okay, so you were right, 1 chiral center there. Let's move on to the larger one. This one has 6 carbons, not just 3. So this is glucose. So looking at glucose on the left, can you tell that it is a hexose? You should be able to count the carbons and to confirm that it is 6. We have 6 carbons there. And it's also an aldose. That's all. You see the top carbon, this is an aldehyde. It's another way of writing an aldehyde. It looks just like this one, right, where it's a C double bonded to an O with an H on it. It's often written CHO because this H is on this carbon and this oxygen is double bonded. It's a shorthand notation for aldehydes, and you may have seen this before, right? So don't get confused. This does have a double bond in it, right? So how many of glucose's carbons, of the 6 carbons here, are chiral? Right, let's talk about each one. Is the number 1 chiral, the top one?
No, because there's a double bond.
Correct. It's not because it has a double bond. How about the last one?
There's an extra hydrogen.
Right, there's 2 identical things, so 2 hydrogens, so it can't be chiral. How about the 4 in between?
They are because they only have 1 hydrogen on each of them, and they have different lengths of carbon chain on either side.
Right, so the requirement is not to have a hydrogen. Don't confuse that. If this was something else other than OH, or if this was a fluorine, it would still work. But as long as 2 things don't match, right? It doesn't have to be a hydrogen. I know the examples we do often have hydrogens there, but it doesn't have to be a hydrogen. But you're right in the other sense that everything attached to this is different. So we have 1 carbon up here, we have a much longer carbon chain down here, we have an H and an OH. So definitely chiral. Even this one, it has a 2 carbon chain up here, right? A 3 carbon chain down here, and they're different from each other in other ways because it had chiral centers on each side of it, and an H and an OH. So all 4 of these have that pattern, so all 4 of them are chiral centers. So how many versions of this are there? We said glucose was an aldohexose. How many aldohexoses are there?
16?
There are 16, right? Because I double the possibilities with every chiral center. So if there was only 1 chiral center, there'd be just 2. If there are 2 chiral centers, there's 4, 3 makes 8, and 4 makes 16. You double it every new chiral center. So glucose has 15 cousins, right? So they look like glucose, except they just vary in how these are attached, right? The OH is on one side or the other. This is a projection that we're naming. I don't care if you know the name of it or not, but it's a way of drawing glucose such that left and right mean something. You notice I drew the OH on the right here and the H left. Here the OH was left and the H right. Why did I draw that differently? And it means something to be on left or right. And that's what we mean by D-glucose. So the last chiral center in glucose, which is which one? What carbon number contains the last chiral center? This is carbon number 1, obviously.
5.
So carbon number 5, so 1, 2, 3, 4, 5 contains the last chiral center. Carbon number 2 contains the first. So as we go along the molecule, the one farthest from number 1 is called the last chiral center. And that's what determines if it's D or L. Right? And that's just the way it works in physically. That's what determines which way it rotates the light, left or right. So
I have a question.
Uh-huh.
Are you recording this?
Am I what?
Recording. Remember to record
I should be.
the session. Thank you.
So if you look at the last carbon, right, that's chiral, that determines if it's D or L. So looking at the last carbon, where is the OH? Is it on the right or the left? And so last chiral center, number 5 here, is the OH on the right or left?
Right.
Right. So we're going to call
Right.
So we'll call it D. If the OH had been on the left, we would call it L-glucose. Right. So that's
The D
what.
is left-handed, right?
Say again.
D is left-handed, though, right?
D
So
means
then-
right-handed. Dextro means right-handed. Right. L means levo or left-handed.
Gotcha. I have one quick question. Why is the double bond not at the top? I know that you said it was, but if I saw that, I wouldn't necessarily. It might trick me.
But CHO is a shorthand notation to write an aldehyde like this.
Okay.
So if you see CHO, though this carbon needs 4 bonds, one of them is to number 2 here, right? One of them is to the hydrogen. That leaves 2 bonds must go to the oxygen. So it is
Okay.
double bonded. It's just a shorthand notation for drawing an aldehyde. Right? So the D stands for dextro and L stands for levo. And an easy way to remember that is, what do you call someone in the modern day who is able to use either hand to do something, to write or to whatever function they're doing with their hand? They're equally,
Ambidextrous.
we call it ambidextrous, right? And do you think of that as a bad thing?
No.
No, it's kind of lost all of its negative connotations. It used to be a derogatory term, right? This was hundreds and hundreds of years ago. So when someone was ambidextrous, it meant exactly what the words mean. So dexterous means right-handed, right? Levo meant left-handed, or sinister meant left-handed, because they thought left-handed people weren't right. And then if you say you're ambidextrous, ambi means ambiguous. So what it meant was if someone was ambidextrous, it literally meant someone was unsure which is their correct hand. Like they can't even figure out to use their right hand. So it was a demeaning term. We don't use it that way today. It's lost all that connotation, and now we mean to literally mean you could use either hand. Right? But it's lost its original connotation. So that's where this word comes from. Dextro means right, right? And levo or sinister meant left. Okay. But glucose is an important sugar. That's why we're going over it. Also known as dextrose, right? Often called that for that reason. Grape sugar or blood sugar. And it's normally found in the blood at about 70 to 100 milligrams per deciliter. And for some reason, the medical community has decided on this very odd unit, probably because it tops out around 100. But a more realistic unit to use, which instead of these, no one uses deciliters outside of medical profession, is to use something like milligrams per milliliter, which would be even more convenient, I think, because it ends up at 1. Right? So if it goes over 1, it's a little high. If it's below 0.7, it's a little low. So if you're below this 70 number, or 0.7 on this scale, you're called hypoglycemic, or a low blood sugar. And something above 120, 130 is called hyperglycemic. Right? So if it's above 130 or so, your blood sugar is high. Now it does vary depending on what you're doing. Like immediately after you eat something, right, especially if it contains an appropriate amount of carbohydrates, then your blood glucose concentration will spike, right? So it goes up to roughly 130 or so, and then gradually falls over time as your body releases insulin and absorbs all the sugar from the blood. So when you're taking someone's blood glucose concentration, whether it's through an arterial stick, which is sort of painful, or just in a regular vein, which is not so painful, although children will disagree with you. It's killing them, of course. But when you take that blood glucose, you got to remember, did this person just eat? Are they sitting there with a glass of Kool-Aid in their hand while you're taking their blood sugar and it comes out to 136? Don't be surprised, right? But if they said, no, I haven't eaten anything all day, nothing but water overnight, I was preparing for surgery, and you're ready for that next morning, you measure their blood glucose and it's 75. Don't get alarmed that that's low. That's normal because they haven't eaten anything in a while. Right? So you have to put it in context. So don't think because you get a blood glucose of 75 or 110 that it's abnormal. Okay? Let's compare two sugars here. So we have glucose on the left, right, and galactose on the right. It's a cousin of glucose. One of its 15 cousins, right? So one of the other ones I'm going to ask you to know. The other is 14. I don't really care about if you know it, because these are the important ones. So glucose and galactose. Okay, so glucose differs from galactose. How? What is the only difference in these two molecules?
The OH and H on the fourth carbon are switched.
Exactly. So the OH on carbon number 4 is on the right for glucose, but on the left for galactose. That doesn't change it. It's still D-galactose. It's not R or L or anything else. Number 5 is still to the right. The last chiral center is still to the right, and that's going to be true for all the sugars we talk about. Okay? But one of the other 3 has changed. So this one's on the left. And that is the only difference between glucose and galactose. So instead of having you memorize a new sugar like galactose, I want you to memorize glucose. If you can draw glucose, and it's easy to remember because all the OH is on the right except number 3. Right? The second OH, or the one on number 3, is to the left. If you want to remember it as that way, or as marching orders, like, it's right, left, right, right. Right? 2, 3, 4, and 5 are right, left, right, right. Right? And the last one is never chiral. The first one is not chiral because of the double bond. So if you want to remember it, however, be able to draw glucose, because if you can't draw glucose, we're going to have trouble with the rest of the semester. It's very dependent on you being able to visualize this. So if you draw glucose and memorize it, you don't have to draw and memorize galactose. Just remember the only difference. Just draw glucose again and then switch number 4 and you have galactose. So think of galactose as a footnote in your learning of glucose. So when you learn glucose, you draw this, and then you just remember to change number 4 around, and you have galactose. And that's true for all these cousins of glucose. They're just one or more switches of these OH groups, but the only one I want you to know is galactose. Okay, so just switch number 4, and you've turned glucose into galactose. And that's the only difference in these. Okay, so fructose is quite different from glucose, right? It's not one of those cousins because it's not even an aldose, right? It's actually a ketose. You notice the double bond is down here at number 2 instead of up here at number 1. So how many chiral centers did glucose have again?
4.
How many does fructose have?
3.
Only 3. Right? The number 1 is still not chiral for a different reason, but it's got 2 H's now. Number 6 is still not chiral for the same reason, it's 2 H's. And number 2 is no longer chiral because it's got the double bond. That only leaves numbers 3, 4, and 5 as chiral. Okay, so when I go from an aldose to a ketose, there are fewer chiral centers because number 2 cannot be chiral anymore. Right? So in general, the ketoses have 1 fewer chiral center than their equivalent aldose. But if you look at glucose and fructose, let's say from the shoulder down, right? So from this point down, they are identical to each other, right? So the number 3 is to the left, number 4 and 5 are to the right, number 3 is to the left, number 4 and 5 are to the right. So glucose and fructose from the shoulder down are identical. They just change their head and neck around, right? The double bond is instead of on 1 is on 2. Right? So these are isomers of each other still, as is galactose. But this is a ketose. These are aldoses. Right? So you don't have to remember fructose as a new sugar. You can memorize glucose like you did before and say, what's the difference? I just move the aldehyde down to number 2 and make it a ketone. And that leaves 2 hydrogens on the first one, so it doesn't matter where you draw the OH because it's not chiral. Right? So again, like galactose, fructose is not another sugar you need to memorize. It's simply an alteration of glucose. That's the way to learn it. If you try to memorize it as a separate sugar, you're taking the long way. So just memorize it as I just moved the double bond to number 2 from glucose and I have fructose. And fructose here is the sweetest of all the sugars, known as fruit sugar. We have a receptor for fructose that we perceive as sweetest of all the sugars. Right? Glucose is the main component of things like honey. Right? It's sweet, but not nearly as sweet as the sugars we get out of fruits. And that's to attract the animals so we can spread their seeds, of course. Right? So here's another sugar I want you to know, and it's ribose, and it's very closely related cousin, deoxyribose. Okay? It's different from glucose in that it only has 5 carbons. Right? So glucose, again, had 1, 2, 3, 4 chiral centers. Ribose here has how many chiral centers? It's an aldose, and it's got 5 carbons.
Three.
The top one, again, is not chiral because it's got a double bond. The last one is not chiral because of the 2 H's. But the 3 in between, just like glucose, are all chiral. The easy way to remember ribose is it's only got 5 instead of 6 carbons. And as the name implies, all the OHs are on the right of ribose. Every one of them is on the right. So you don't have to remember an odd one to the left. Right? And the difference between ribose and deoxyribose is pretty simple. There's a deoxy, a loss of oxygen at number 2. So this OH that's normally on number 2 is not there. Right? So ribose, you said, had 3 chiral centers. How many does deoxyribose have?
2.
2, right. Only the carbons 3 and 4. Number 2 is no longer chiral, right? Because it's got 2 of the same thing. And we find these important molecules here, this ribose and this deoxyribose, in nucleic acids, right? These are our sugars that make up the RNA or the DNA in our nucleic acids. Right? That's why they're called RNA for ribose or DNA for deoxyribose. Right? So we'll talk about those briefly later when we get to those molecules later in the semester. Right? So the last thing I want to mention here is some of these, in fact, almost all of these, are cyclic monosaccharides in one way or another. If I take a straight chain molecule, sugar, let me show you a picture of one, like glucose here, right? What 2 functional groups does glucose have? It's got many of one type, but only one of another. So what 2 functional groups does it have?
Hydroxyls and carbonyls.
So carbonyl in the form of an aldehyde, right? So it's got an aldehyde group and a hydroxy group, exactly. Can you think of a reaction between an aldehyde and a hydroxy group? We've done this before.
Oxidation.
So aldehydes and hydroxy groups can be oxidized, that's true. And the aldehyde group could also be reduced, that's also true. What other functional or what other reaction could occur between them?
Hydrolysis?
Just the opposite. Not hydrolysis. We're not going to tear them apart. I'm going to put them together.
Dehydration synthesis.
Almost. Not quite yet. So last time we talked about putting an aldehyde and an alcohol's hydroxy group together. And that was the formation of hemiacetals. Right? And that's exactly what we're going to do again. So this hemiacetal, right? I don't have a picture of it here for you, but a hemiacetal, we've done this before. We take an aldehyde plus an alcohol, and you remember what happens? You have the carbon here with its carbonyl double bond becomes a single bond, right? And then this carbon makes another bond to the O of the alcohol, right? And then the O here, which lost one of its bonds, is going to get another H on it, right? And then the alcohol's OH is now connected to this carbon. So I want to use this glucose here to describe it for you. I would normally demonstrate this in class with a little silly dance, but we're not in class, we're not in the lecture hall. But imagine this molecule is head to toe, right? So the top is its head, we have neck, shoulders, waist and knees, and then feet down here, right? So imagine it top to bottom, it's standing up, a glucose here, right? And so this OH here at the bottom, the last chiral center's OH, right? So this is kind of like its back of its heel or its ankle or something like that. Not quite the toes down here. So we get to this point, and this molecule is going to kick itself in the back of the head, right? It looks sillier when I do this in class, right? But this O here, this OH, is going to get involved in a reaction with this aldehyde, right? And the way I do it is it comes around this way. It has to come around this side, right? So this O attacks this carbon, right? Makes a bond to this carbon here, so new OC bond, right? This H is lost, right? It's no longer on this O. It's going to end up on this O, and this double bond becomes a single bond. So what will this top carbon look like now? It won't have an aldehyde anymore, but it will have an OH, still got its H, still got the bond to number 2, and it'll have a new bond to the O of number 5. This will then be in a ring, right? You see how if I make a new bond between this O and that carbon, it'll be in a ring. So I'll have my sugars, what's known as their ring forms, right? Or their cyclic monosaccharide forms. So it's a ring closure, just like we did before for the hemiacetals. It's no different than the others. I'm just doing it in the context of a sugar. And it'll always be the last chiral center's OH that's involved. None of the others. And always the aldehyde, or in the case of fructose, the ketone. Right? So in the case of fructose, instead of kicking itself in the back of the head, it kicks itself in the shoulder, because that's where the carbonyl is. This will make a smaller ring. So I want you to take these sugars, the glucose, the ribose, and the fructose, and if you want the galactose as well, and make their ring forms on your own, right? And then look up what their ring forms look like. We'll talk about these after class as well. Right? But take these sugars and make them into their ring forms and see that you're making a hemiacetal. Okay, so try that, and then we'll talk about it again once you've accomplished that next time. Right, and then the last thing I want to mention is the term called reducing sugars. And you've seen this before when I showed you the Tollens test and the Benedict's test for aldehydes. Remember the Tollens test, the silver mirror test, that reacted with the chemical if it was an aldehyde and precipitated the silver? Or the Benedict's test that reduced the copper from a blue solution to a brick red or orange solution? Right? Only if an aldehyde was present. So if a sugar like glucose shows up and it's in that reaction, and I think you guys will do this in lab, right? So if it has an aldehyde group and you react it with the Tollens or Benedict's reagent, then it will react, right? And what happens is it oxidizes this, right? So this aldehyde will become a carboxylic acid, which we'll talk about more next week. But then the metal, the silver or the copper, will get reduced to its metallic state, and you see the color change, or the silver deposition. But all aldehyde sugars will react like that because they have an aldehyde group. So all aldoses will test positive in the Tollens and Benedict's tests. So if you're testing for reducing sugars, right, a reducing sugar is able to reduce the metals in the process get oxidized, right? Then all monosaccharides will test positive, right? All the aldoses will test positive, and in fact, all your ketoses will test positive, even though you know a ketone cannot be oxidized, right? I'll leave the mystery of why that happens for your lab instructor, but all monosaccharides, no matter what they are, aldose or ketose, will test positive as a reducing sugar. The ketoses have to go through a separate step, which your lab instructor will hopefully explain. Right? But the other case with the aldehydes here is we can take an aldehyde, and instead of oxidizing it and it acting as a reducing sugar, we can actually reduce it. So we take this aldehyde, and aldehydes reduce to primary alcohols like this. So we use some reducing agent like H2 and a metal catalyst, and we reduce it to a primary alcohol. This is no longer a sugar because it doesn't have the formula for a sugar anymore. It's saturated, right? And it's called sorbitol. And it's not really as sweet as the original sugar. It's sort of sweet. It kind of binds to your receptor, but not very well. And this is what's used in what they call sugar-free gum. It's not so sugar-free after all, because your body can oxidize this sometimes back to the aldehyde, and you actually have sugars present. So they use this in these sugar-free gums because it doesn't contain any sugar, but it does contain a modified sugar, right? It's called sorbitol because it's just an alcohol now, right? It's a hexol, right? Does that make sense? You can either oxidize this and have it test positive as a reducing sugar in a Benedict's tolerance test, or you can reduce it and it's technically not a sugar anymore, and they use it in these sugar-free gums called sorbitol.